Good morning, everyone. Thank you for taking the time to join us today for this exciting webinar brought to you by Syntica. My name is Ivy, and I will be your host for today. And today we are joined by the renowned Dr. Timothy Haystead. So today's webinar is titled Targeting HsP90 and its Pathogen Orthologs with Tethered Inhibitors as Diagnostic and Therapeutic Strategy for Cancer and Infectious Diseases. I am pleased to introduce our featured speaker, Dr. Timothy Haystead. He is a professor of pharmacology and cancer biology at Duke University. Dr. Haystead earned his PhD in biochemistry from Dundee University in the UK and completed his postdoctoral work at the University of Washington in Seattle under Nobel laureate Edwin Krebs. Dr. Haystead started his independent career at the University of Virginia in 1991, where he focused on understanding protein kinesis and phosphates signaling pathways using small molecular inhibitors. His innovative work led to the creation of Pure mining platform, which eventually led to the founding of his first company, Theranostic Incorporated, in Durham, North Carolina. In 2000, Dr. Haystead moved to Duke University. Theranostic used Dr. Haystead's platform to discover and develop the first fully synthetic HsP90 inhibitor, which entered clinical trials in 2008. After Pfizer acquired Theranostic that same year, Dr. Haystead continued to innovate in developing HsP90 as a target in creating highly selective inhibitors of purine utilizing enzymes. This work has resulted in novel imaging probes and methods to selectively kill tumor cells using tethered molecules. Dr. Haystead's current research focuses on developing these advanced tools as theranostics, PET agents for cancer and infections. His second company, eHsp90, also based in Durham, North Carolina, has licensed one of his molecules from Duke to target TNF-mediated signaling. So before we begin, I have a few housekeeping items to cover. This presentation will last about 45 minutes, followed by a Q&A session at the end. Please submit your questions in the Q&A box throughout the presentation, and we will address them at the end. We will provide a transcript of both answered and unanswered questions after the session. Additionally, today's webinar is being recorded, so if you lose connection or miss anything, you can review the video later. And without further ado, I am pleased to turn this over to Dr. Haystead for today's webinar.
Hello, everyone. Thank you for the kind introduction. And I'll just get right into it. So from my introduction, you'd see our history in developing small molecule inhibitors. Over the years, this has led us down the path that we're currently at, which is now to try to develop novel theranostic agents for imaging signal transduction proteins, starting with that HsP90, but I think some of the methods that we're developing could have broader utility for many other interesting targets. So this is the crystal structure of HsP90. It's the yeast form with one of our tethered inhibitors bound, and deep in here would be the active site with a ligand on it. We're going to hear about those. There's a tether, which carries an imaging modality. This is now hopefully going to be a PET agent. Now, we are new to the PET field, and we're learning. I am surrounded by experts in medical physics, and I'm also married to a radiologist, which helps. So we're hopeful to be able to take our small molecule platform technology and advance it into signaling. We do have some PET data that I'm going to show, but it's really at the cellular level. But I'm interested in hearing from anyone in the community about how to do this. Just to give you a little bit of background on my expertise, we mentioned the company we founded in 2000 called Theranostic. It was in a garage. It was based on a platform that I use routinely in my labs to this day. It's proven very fruitful for the development of novel ligands that are highly selective, and that's obviously a critical element in the development of any drug, but even more so for any imaging agent. You need to make sure you hit the target. The other part of the equation is the target's biology obviously must be relevant to disease. In cancer, we picked this target HsP90 many years ago. It's an unusual target, but it's also a target that I think is of great value, particularly in cancer and also in infectious disease, which I hope to give you some sort of flavor of. We published pretty heavily on this target over the years, and we've actually progressed 3 molecules to the clinical level, 2 which have actually entered clinical trials. The latest being a novel imaging agent called HtpG, which is a histological tool. We'll touch on that. But really, we want to get into the being able to image this target in the human body, and because of its connection with cancer, and also it turns out to be a great target to move into infectious disease. We're sort of funded for both areas, sort of weirdly. This is corporate headquarters, original corporate headquarters for the second company called eHsp90 that we spun out recently. We now have an actual office to develop this kinase inhibitor, and this was our company, was in a garage in downtown Durham for about 8 years. So you can see we kind of have a low budget on where we do our science, but the science itself is very sound because it does deliver things like drugs and things that go into people. So this is the platform that we use that I'll touch on as we go on. We've used it routinely. It is a platform that is rather unique in that it enables us to interrogate a lot of enzymes at once to define what we call chemical starting points that can then be developed into selective ligands, of which HsP90 was just one example. It's based on an early patent that we got on back in the early 90s on immobilizing ATP in a specific orientation, such that when it's bound through its gamma phosphate and held on a resin, it will bind about 2,000 proteins in the human genome, or about 10% of any genome. That includes pathogens. Reversibly, nature orientates ATP such that these phosphates stick out to the solvent accessibility, and the enzymes will bind all the way around ATP. You might ask, well, how could you ever get selectivity? It turns out because of the 32 kilodaltons of amino acid sequences required to orientate ATP in this way, there's a lot of sequence variation that makes it possible to discriminate one of these in the human genome 2,000 things from each other. So the idea is that you mix a biological milieu with this resin, and then you interrogate that milieu on the resin against a small molecule library of drug-like molecules that you can apply all kinds of rational design, AI, or whatever you want here to develop your library. You pass it through, and you look for things that get eluted selectivity. So the assay not only identifies promising starting points, but it also can be used to get a feel for their selectivity, which is the most key thing. And here in this is a silver stain gel that was recently done. It's a pathogenic yeast we were looking at with some colleagues at Duke. We're looking for scaffolds that based on our HsP90 libraries that target the protein HsP90. This is the yeast form in this case. And here are 2, one called HS10 and one called HS289. This is a Bayer compound. This is my compound developed by the theranostic company. But it selectively elutes just this protein. It's obviously been an optimized ligand, but not all these other ones, which are, if you use mass spectrometry to sequence what sticks to the media, what remains on the media, or what's elutable with ATP, you get this long list. This is a sample. So you can see the specificity there. It's real data. You can see what keratin's in the gel, which is bad handling on my part, probably. We have a number of, over the years, been funded for all kinds of discovery. We're going to talk mostly about our HsP90 program, and these are the indications that we've gone into. We're going to add pathogens here now, bacterial ones and yeast, probably parasitic ones we've published on as well. And then a number of kinase targets and things that we worked on in a year using this platform that has led to discovery of novel selective ligands that are very selective within what we call the pyranome, which is all these things that bind ATP. We published on these very extensively. You can see where we made most progress, but a number of publications out of my own lab. Our list is quite extensive because we often share these with others, and so that gives you some idea of that the robustness of some of the ligands we're discovering, and we have a high degree of confidence that the ligands that we make are very selective for the targets, and they're usually very potent. So hopefully we can convince you on target selectivity. We've recently got into these tethered versions, and I'll describe how we make those in a second. These are ligands that have a PEG arm on them and will carry something like a payload, which can be a poison. This is burgdorferi, or it could be a fluorophore, which you'll see. And we are now currently making things that carry metal isotopes for PET, etc. So we have increasing numbers of papers that we published in peer review on those. So how do we do all of this? Well, typically, I'm a biochemist by training, so we do lots of biochemistry first. This is an example. We have the same assay that we use to discover these ligands with vast numbers of diverse enzyme classes. You can look at all of them with it. HsP90 is a particularly difficult enzyme to assay, but we use a simple binding assay based on its interactions with ATP without affinity media. We express a protein as a GFP, and then we can titrate that protein. These might not look so potent to some of you, but it's actually done in the context of extremely high ATP backgrounds, which forces selectivity. It's done at 5 millimolar ATP. The KDs are typically in the low nanomolar for those that are into that sort of thing. But in this particular study, what I'm going to show, and it's an example of how we go about trying to derive a selective ligand, not only within the human pyranome against all other proteins, but we've really got into this idea that the scaffolds that we developed for the human program can be used to target bacterial and yeast proteins and some parasitic proteins like in malaria, and these libraries we created around HsP90 are useful starting points for pathogenic protein HsP90 orthologs. And this is an experiment where we're targeting a Lyme disease ortholog of HsP90 called HtpG. And you can see these particular two ligands here. My scaffold is not so good at inhibiting this enzyme, but a Bayer compound called BXH289 turned out to be a great starting point for that. So we began a process of trying to understand this ligand, which is a known HsP90 inhibitor. And you can see they're very similar to the human, but show this differential against this bacterial version. So we use that, and we got a co-crystal structure of that ligand. Bound. This was done by Matt Redemba, my good friend and colleague at UNC. He quickly found a crystal structure of the bacterial version. Here's our original crystal structure of the same protein, same molecule in the human form. You can kind of then overlay these two things and understand the amino acid contact differences between this starting point. And what we're trying to do here now is try to create a ligand that dials out the human form completely, but retains the bacterial form. And we use this ATP binding assay to do that, simply because why not use an ATPase activity, you might add. That's a difficult radioactivity assay. There's all kinds of nuances why you want to do that. But HsP90 is a difficult protein to assay in high throughput because it is involved in protein folding, and nobody really come up with a good assay. So we use a simple binding assay to do that. So once we start to understand the contacts between this ligand and the target protein, we can start to make iterations through an SAR process to develop very specific inhibitors. And this is the nature of the assay again. you typically take a GFP fusion on expressing the bacteria. So we're not looking at just native extracts anymore, but we're looking at just this one protein bound to the ATP resin, and it's green, it binds really well. You can see it, quite these resins grow very green. Then you start to interrogate it. In this case, we're going to interrogate it with a sub-library from our HsP90 inhibitor, and we're trying to look at for like selective inhibitors. We do the same thing with a human, and we're looking for signals that are eluted specifically from this with this protein and not the human counterpart. And it's a very robust assay, and your signals really do look like this, actually. So, and it's a very fast assay. So to sort of cut along chemistry story short, you can see by these numbers, we went through some 300 plus analogs to interrogate both the affinity for HsP90 and the affinity for the bacterial protein. And we typically will design, break the lead into parts to do this iterative process, making substitutions to try and dilate the human, but retain binding to the bacterial form. And you can plot one against the other, and you get this sort of interesting plot, and you see things start moving away from the human. They get worse KDs, but retain potency for the bacterial version. And to cut a long story short, after a while, we do arrive at a target separation. It's quite impressive, at least in our assay, and actually in other assays, more biophysical ones that my colleague does, in which we've completely dialed out the human orthologs of HtpG, the HsP90. So we now, if we want to image this HtpG, which we're moving towards, and you'll see a little bit of data where I've got some promise there, at least at the cellular level, we can separate them. So this same process we use not only for HsP90, and it's works rather well, we also use for other proteins outside of HsP90 now. So just a second, and get my timer back here. So you can see that we've created a ligand specific. We have ligands that are specific for human. We have ligands specific for this particular pathogen. We can actually dial out other pathogens. We started to work on this. You can see in this particular example is the amino acid sequence of Borrelia HtpG compared to its closest homologs in the bacterial world. These are all spirochetes, and we've even started to use the process to dial out other species that might be of diagnostic relevance if we're looking at pathogens in infected animals and humans. So you can play with these amino acid differences and with the power of SAR and molecular modeling and come up with an inhibitor that will most likely just be specific for one pathogen or the other, some of which we have actually published on recently. Just to give you some idea how clean these are, this is the HS463, which is our Borrelia specific or spirochete specific, doesn't bind human. If you make an affinity resin out of it, put a tether on it, the tethered version still binds the target with reasonable potency. But you can, a classic affinity experiment where you mix it with extract, you just get the target recovered. I keep laboring this point about specificity because I'm sure all of you that are involved in molecular imaging or using imaging agents, you want to be sure at some quantitative biochemical level that you're targeting a protein of interest that is selective and doesn't target other things. And so I believe we've got quite a solid methodology that enables us to achieve that goal. So what we've been able to do with this, we talk about tethered inhibitors, and I'm going to show you some of our cancer data right now, since we've taken that the furthest all the way to clinic, to the point where we really do need to make a PET version of this target. Because the other half of the equation, obviously, you've made your ligand, you've biochemically convinced everybody that the ligand is specific. Now you want it to carry an imaging probe or a poison or whatever if you want to target, but there must be biology there in place that you can do this selectively. And it turns out HsP90 does have some biology. I'll convince you in a minute, even though it's a ubiquitously expressed high copy protein in all cells. It has an actual form that is associated with metastases that appears on the surface of cells. But we were able to find this form. We're taking our HsP90 ligand. This is the SNX scaffold. They talked about HS10. And, we put make an affinity resin out of it, and we published on this. You can show it just binds HsP90, doesn't bind very well the bacterial forms. And but then once you've done that, and you've now got a delivery agent potentially, you can attach fluorophores, do your cell biology, which is essential before you go to the more interesting bits that you guys are no doubt interested in, which is the things that carry chelates or other PET moieties. So you can see in human disease, the role of what this protein is playing and perhaps target it. And we've actually made everything that's on here. We've done a lot of work with fluorescent ones. We've done a fair bit of delivering poisons to kill tumor cells and actually now bacteria. And we're really focused around here now, trying to translate this into a way to deliver agents. And we use tethers in this case. You'll see why we need to do that in a moment. We can also not use a tether and attach a PET agent directly, although you have to be careful where you put that with these, because the electron crowd associated with things like iodine actually affect binding quite significantly. Chelates obviously will need to be tethered, something added to them, so you can add a metal, and we're looking at both options. So to get to the biology and why this protein HsP90, the most unlikely target one could possibly imagine, but it does have some advantages in PET imaging. It's a very high copy protein, for one thing. It has interesting biology. That's the other thing. A close colleague at the NCI had originally published a paper that suggested there was a surface form of HsP90 associated with metastases. And he'd made antibodies that he showed in culture would inhibit HsP90. So we thought we'd look at this, and we made a ligand that had the tether on it, and we put an imaging agent on it. What was interesting, we chose fluorescein because it was known not to enter cells. And sure enough, this ligand will not enter cells unless it's trafficked in. But on its own, it won't enter cells. A normal drug will diffuse freely. Now, if you had a PET labeled version of an HsP90 inhibitor, and this was actually done using another molecule called a PUH71 with iodine on it, it will diffuse everywhere in the cell. You do end up getting contrast eventually as a PET agent using the iodinated version, but it took about 2 or 3 days before they saw contrast. As you'll be convinced, hopefully, our agent will bind to a surface form of HsP90 that Leonard described. And not only that, it will bind, it gets internalized, and it will accumulate to quite high levels. And this turns out to be an advantage for detecting the presence of the surface form, which is associated with metastases. So one can imagine that one could target that protein. So again, it's got a ligand, a tether, and an imaging agent, and it binds to the surface. This is the first piece of data we got really excited for. This resulted in a very large DoD grant that ended up in an actual clinical trial in men with prostate cancer. I'll touch on that. So in this case, we've got this ligand on one end, and this is a fluorophore. One of the first observations that we saw with the fluorophore version in using epifluorescence is that aggressive forms of breast cancer, these are 3 different examples that represent the subtypes, rapidly uptake these probes, whereas a benign version is not so much absorbing the probe. And then this is just a cell type that doesn't express the surface form, and doesn't take the probe up at all. At the molecular level, if you do high resolution 3D imaging, this is actually with a red probe, I believe, at the time. Yes, it's 131. This was done at Genelia Farms. And we add the probe to these breast cells very briefly. And we look at the surface binding. What we see is these aggregates that form on the surface of these tumor cells. And when you look in detail, you see that they get internalized, and then they become quite regular. And they're first they're in vacuoles, and then they exchange with the probe when it's becomes bound to the intracellular version of HsP90, and we've established that it then diffuses throughout the cell and it's retained for a very long time. And that's simply because there's about 2% of the cell protein is estimated to be the cellular form of HsP90. So this is a mechanism where we could deliver these probes. This is quite reproducible, and it has been these compounds we give out regularly to all kinds of laboratories around the world, and reproduce similar results in many, many tumors. So the mechanism is that normal cells don't express the HsP90 on the surface, and that tumor cells, particularly aggressive forms, do. And when they bind our probe, we based on confocal evidence, the probe seems to cause aggregation and accumulate in what we believe were in a silencing RNA experiment years ago. We found involves clathrin-coated pits. We think one of the functional roles of HsP90 on the surface in metastases is to traffic and chaperone cell surface receptors such as HER2. That's why it's being trafficked. But it's being recycled, and we believe this to be a conserved mechanism that's even conserved in pathogens. So you can see that they get re-internalized. If you inject a mouse with these probes, and just to see where they go, this is the fluorescent version. And in this case, this is a tumor on the side of a mouse. We injected it into the mouse up the tail, which is what this fluorescence is here. And then we look at it at different time points. You can see accumulation of the tumor. That's a necrotic region in the middle. That's the tumor on the flank. The drug is actually eliminated through the bile into the liver and out into the gut. And you can reconstruct this whole mouse actually in 3D, which you can find online. It's kind of cool. But the take-home message was we really didn't see a lot of binding elsewhere. So it would seem that normal cells do not express HsP90, at least in an animal, and do in fact find their target in the tumor. And this is a floor version. Our collaborators that were involved in the clinical trials is some of their work. So we actually made a deep red version. This is called HS196. This ends up in a human study versus an inactive analog, just to show you. And what our collaborators did, they took mice bearing tumors. These were breast tumors, aligned. You got 4 controls and 4 active drug. This is an inactive analog we made. And you can see the inactive analog is quite clear quite quickly, whereas the active one is retained in all the tumors quite convincingly, even was detectable in 168 hours. So it has a long half-life. Again, ticks a box for potential imaging agent and a mode of treatment. To really cut a long story short, this ends up in a clinical trial in men at Duke University, and this was also published in peer review. These men had aggressive form of prostate cancer. They had been through multiple treatments, but so they were earmarked for tumor resection. They volunteered to take this probe, which we took all the way through IND. A very small dose per person, and this is one of the results. Is the tumor? The tumor was sectioned and then imaged. Along the way, in a blinded way, a pathologist at Duke, Yohiti Wong, had carried out his normal path analysis, and then that analysis was cross-referenced with this analysis, which is the fluorescent regions. In his eyes, all corresponded to a aggressive disease in these regions of the tumor. Single cell sort was done on these, and you can read this. It's really fascinating. What you find is the fluorescence correlated with cells with a neuroendocrine phenotype, which is a very aggressive phenotype. So this is one of the first pieces of evidence that we've got that in vivo in a human being, our probes will find their target and will light up the tumors, quite specifically the aggressive forms, which opens up a big window for trying to validate this target more broadly in other tumors. We know it's expressed in many, many tumors in humans. So it's probably worth really pushing this to join a rather short list of molecularly targeted ligands that are in clinical practice. There are two that I can talk about. This one is sort of interesting in that it would have very broad utility. Those other two are limited to PMSA positive prostate tumors and the somatostatin receptor. I think we're in the audience poll right now.
Thank you, Dr. Haystead, for the fascinating presentation thus far. We're going to take a quick break for our audience to learn more about you guys and what your interests are. So we have 4 poll questions for you today, all single choice. The first question: what type of animal imaging are you currently performing? Is it PET CT, SPECT CT, ultrasound, optical, influorescent, MRI, DEXA, X-ray, photoacoustic? And our second question is, do you work in vivo or in vitro currently? Yes or no? And our third question: what is your experience with PET CT? Do you have no experience, minimal, some experience, moderate, or extensive, or are you an expert? And we'd like to know what your main focus of work is. Are you in oncology, neurology, cardiology, theranostics, physiology, or something different? So I'm going to give you a couple more seconds for you, the audience, to answer before we dive back in. We're going to give a couple more seconds before we dive back into the presentation. If you haven't already, please answer as many the poll questions as you can. Thank you all for taking the time to participate in the poll questions, and I will hand it over back to Dr. Haystead.
Okay. All right. Okay. So let me get rid of this top thing here. I'm going to go away. Oh, never mind. So I've talked about the ligands and tethers that we've made in some of the imaging studies we've done at the molecular level, which is really set the stage for developing PET versions. We're choosing PET because of its sensitivity. It's probably about the closest thing to the sensitivity one will get with fluorescence. And this sort of shows the whole story of our PET efforts so far. And we're looking at iodine as a source for imaging. We've actually made this molecule, and I've got a little bit of PET data on that in an animal that was done originally with UNC. It's a very preliminary study. We started working on these chelates and run into a bit of trouble with them. It's interesting. But we have a number of options for us to be successful in developing a PET version. Like I mentioned, we are new to this. Our first study was done actually quite some time ago at UNC using their PET facility with the same instrument we just bought from Syntica. As a result, but just to give a sort of flavor of where we're headed, hoping to repeat this data. this is some PET imaging of a mouse that was bearing a flank tumor that was over here. You can see it with the FDG PET. It's the same. This is an animal was later images with our probe, that one I just showed with the iodine on it. And you could pick up the FDG PET the tumor, but what's good about this is that the FDG PET obviously is taken up in all the metabolically active tissues, kidneys, heart, lymph nodes, brain, etc. So can pick up tumors, but obviously anything in these areas could be difficult to discern, and they're certainly not targeted, so you couldn't deliver a probe that has an alpha emitter on it, etc., to kill the tumor. That caused some serious problems. So we all know it's eliminated through the bladder, and I think this is actually desirable and something we need to focus on. It did pick up a secondary mass. This is a spontaneous tumor model, which is why we chose it. It's actually not thought to be any more a relevant model. Typically, the cancer field wanders in and out of what's relevant and what's not, whether it's PDXs we should be using, etc. But as a drug developer, we really focus on a model that will just demonstrate when our probe actually works in a living system, and that's good enough for us. Because the next stop is to try to move this down a clinical path. We can get that data to show the FDA, which is what we did with our floor versions and poison carrier versions. And what's interesting about this, we inject this HS113, which looks like this. It's got fluorescein on it. That actually stops it going into cells through diffusion. It means it's going to be carried in. It had this benzalmine with an iodine on it, which was 124. We worked with Michael Zalewski at Duke, who helped us make this, or at least iodinate it from our stanolated version. And there you can see, interestingly, in the first experiment, in one mouse out of 10, I have to say, which because this tumor model is rather difficult to get secondaries, there was radioactivity in the tumor, but it wasn't anything like here. But it did pick up this secondary mass that, when on histological examination, did actually turn out to be a MET, which is very exciting. What's also interesting about this study is if you see, there's no probe really anywhere else, which is one of the big amazements of this study, and we hope this holds up. It's certainly consistent with our floor studies in whole mice, where we sectioned the mouse and looked at in sections by histology, etc., and some of the work by our colleagues looking at the biodistribution. It is eliminated exclusively through the gallbladder in a single pass, and it went into the bowel. This may or may not be desirable for an agent. Perhaps urinary elimination is more desirable. Need some advice on that. You could obviously clear the bowel of the signal and still have all this region to play with. But the T-half was very short. Within 12.5 minutes was the T-half. So in that time, it was able to find the tumor. So that's consistent with our potency. So all good things. So where are we on it? So one of the things that we're really interested in is different means. So iodine is on the table as something we could use. It's got a long half-life. Obviously, in clinically, we don't really want a very long half-life PET agent, so gallium-68 is more desirable. So we've been playing with some DOTA-based chelates, and the first thing we saw was these do not go into cells at all very well, and they were terrible, in fact. And others have published on this, and we're using cerium-134 here. We got from this is a 2-day half-life from the DOE. And this was pretty consistent. It was also true in the bacterial studies that we get. And we think it's because of the architecture of the chelate, which is rather bulky, as a 3-dimensional structure. My chemists liken it to trying to pull a balloon underwater. That's the idea for their explaining these. And I think there's literature out there that showed with other small molecules, particularly PMSA 11, there was a study published that showed that they got very poor uptake, and they now use HtpC, which is a more linear molecule, which we actually have made. Anyway, we did inject this one into mice at the UNC, and we showed that it did have one desirable feature. It was cleared through the bladder rather quickly. So maybe worth pursuing It's also convenient, obviously, that it's very easy to do a mix and go. So you worry about the safety of this or toxicity of the pharmacology of this molecule and its pharmacological characteristics, but you don't really have to worry about the chelation, which is very simple as a mix and go. So we did embark on an idea of trying to make these molecules less polar, and have made a whole series of these that we are currently working our way through. We have not given up on chelates. We do believe that the HbCC version is probably the one to go with. But out of interest, we've sort of tried to shield the polarity of these things and retain the chelate binding. So it's a little bit of a dry subject, but essentially what we're doing is trying to, we keep our ligand constant, we're varying these different chelates, and we're trying to look at uptake in these tumor lines and also in different bacterial versions. Again, our assay comes into play. Every time we make a chelate, we have to validate its affinity for the target. So we do it biochemically. It's very easy for us to do. We're going to HsP90, lots of iterations, so you can prove some of them are a bit of a disaster. They lost all affinity. Here's that bacterial one. You can show it doesn't bind the human with a chelate on it, which is good. We've got two nice binding curves, a little loss in affinity, but still actually in game. These are actually, when you correct for the ATP concentration, as I say, they're nanomolar inhibitors still. They look like this. And we started to play with them in cells. Along the way, we were fortunate enough to raise funding to buy our own PET CT machine, because the first thing you learn is that you have to do this almost every day. You have to do it at the cellular level, before you proceed to animals, and it's very difficult and expensive to get calls to do it. So we convinced our funder, which was the Stephen and Alexander Cohen Foundation, who kindly provided funding for this machine, which arrived about 3 or 4 months ago. And I think we've broken it twice already, much to Synteca's chagrin, but they've been very kind in helping us keeping it going, and taught us a lot. So we have this in-house, and we've started to do some things. Let's call these phantoms. This is where we really are. Obviously, for those aficionados that work on with chelates, we've tried to establish the molarity of binding of the chelate versus the probe. We don't want any free metal. Free metal will go into cells and as a confounding signal, so you don't really want any of that. We endlessly have been doing these sort of titrations. We use set packs to look at, and it's kind of fun to put our titrations. These are 2 different versions into the PET machine. It teaches us about how to use this instrument, but it's rather fun. You can see it all bound to the set pack and how it over the dose response, it teeters away for kicks, and you can actually see it appearing in the breakthrough over here. We use the Imalytics software to do this. It's rather wonderful, very useful, brings these things into alignment really well, and is very quantitative. So we can make plots and things. We can do some interesting things that we need to establish, that we can see the biology with this method. Here we've got a series of test tubes that have had PBMCs in them. If we activate PBMCs with LPS, they will actually induce the expression of the HsP90 protein on their surface. This is another story that came out of work we had with HIV, but the take-home message is you can actually see the uptake in a time-dependent manner into these pellets. So we're actually mixing our probe with a chelate on it, and then centrifuging the cells. And there's about 1 million cells in these little pellets here, and you can see them by CT, and you can do some quantitative things like the volume of the pellet is actually equal to the cell volume. So on the CT, and then you can match that with the signal you get with the PET machine and convert that to becquerels, etc., to look at uptake and come up with moles and things, which we get excited about. But here you can actually see some biology, looking at uptake at the 60-minute time point. The LPS treated ones greatly stimulated in the uptake, so that's quite nice, and that seems to hold up for about 120 minutes. Of course, I was doing the biology and lost the control for this one, but it's still high, and I'm sure there's a control there. But the point is that this has given us a very nice way to try and sort through that big list of chelates that we've been working with. We're trying to calculate what the actual moles is, what the kinetic of the uptake is before we proceed to an animal. But we're zoning in on a candidate, which is probably going to be an HtpC version. This is some latest work. This is actually an HtpC version called H590. It's got a very rapid kinetic uptake. We did a couple of, I think, last week, and again you can see these time points. So you see this uptake into the pellet, see nice uptake in this cross-sectional view. It's kind of fun, and different time points at different points. If you move through, the resolution of the superorgus is pretty exquisite. You can see these things aren't really pointing or even at the same height, and as you scroll through, you pick up the heat. There's actually a similar signal at 30 minutes. It's just see that edge of that pellet there versus a full pellet over here, and the control very short time points. So this is quite an interesting way to look at these things, I think. The other option is to wash these and count them in a scintillation counter, which is rather tedious. So that's our pet story in humans. I'm getting to the end. But we along the way, and where we are right now is to start to look at the pharmacokinetics of these probes in an animal, obviously. I touched a little bit on the iodine versions that are also, we do know, go into tumors, so they may be our option. It's just the iodine installation is a bit more awkward than chelation. But once we've got our kinetics down and we're certain we've got a sufficient copy in their probe, the signal is going to be getting all the math add up with our medical physics colleagues to suggest that we should be able to see tumors in mice in the next coming weeks. We have to do an actual experiment in mice with these, but we're not going to do that until we've got a candidate we know is working at the cellular level. Makes sense. Now, on a bacterial project, which we're also heavily funded for, we started this work in Borrelia using this HtpG probe, the 463. We have made a chelate version of that, and we're working on an iodinated version of that. But the libraries we created for around these now open up the door for all of these pathogens. And we've started to work with various infectious disease people. These are all the pathogens on the CDC wanted list that are problematic for human, where we've got massive drug resistance. There's not much on imaging in infection, so it's kind of a novel area from that point of view. But we think that these theranostic approaches might have utility for these, and we sort of getting quite attractive funding for that to investigate that. So it's the same idea, The idea is if we could image them, we could see where they are, and we could deliver things. Interestingly, just from a biological point of view, despite HsP90 being a very old protein and conserved, the amino acid sequence identities within the ATP binding site vary considerably. And so we think this is a chemical biologist dreamland in making all kinds of selective ligands for different species, which could have utility for diagnostics. This shows you that they do go into Borrelia burgdorferi. This is one carrying the serum 134 again. Now, this is kind of neat, something we played with. These were Borrelia, about 1 million cells again, incubated with our probe for 90 minutes. And then we just simply centrifuge. We don't even remove the media. You can see the excess media at the bottom of these test tubes, but you can still see probe uptake. Working with some of these hot isotopes is quite nice because they don't have to draw that liquid off, and we can still measure what's in the pellet. So that's makes the process even more rapid. But we can see convincing uptake using our Argus instrument into these pellets. So I'm quite excited about this project. Might have feasibility to advance something for infectious disease. It is an attractive animal model for Lyme. You can do in a mouse. That might make it feasible to do some imaging infections in that animal and perhaps advance that for Lyme disease. Anyway, so just to end, like 5 minutes over, I don't do this on my own. I have a big lab that people have worked with me over the years. There's probably been about 30 people involved in this program in my own lab alone. These are some of the current folks involved, past and present, actually. Some new medical physics expertise that are coming in. These are 2 master students. They're going to teach us all about how to handle nukes. Mark Oldham, who's a medical physics professor at Duke, is really helping here. these are all the folks that we've worked with in the past to enable the clinical studies over here. Were funded pretty heavily by NIH and these other organizations, particularly the Cohen Foundation and Bay Area Lyme, helped with the Lyme and the purchase of the PET machine. And we work with other experts that we published with around this program. So there we go. Time for questions.
Thank you so much, Dr. Haystead, for such an insightful presentation. I'd like to open the floor for questions. We have one question. Are there any examples of molecular targeted PET agents using small molecules in clinical practice?
Yeah, interestingly, they are few and far between, given all of the excitement over oncogenic targets and things over the years. But really the best examples now is PMSA11, which is growing in its utility. This is a ligand that targets the PMSA receptor on the surface of PMSA positive prostate cancers. It's quite remarkable in its selectivity for that class. It is probably a true molecularly targeted inhibitor. It's a small peptide that binds to the surface with a tether on it, carrying a chelate. HtpC works. This is our actual yardstick. We're actually bringing that molecule in to give us some measurement of how our HsP90 stacks up against that in the PMSA positive tumor lines. So that's one example. The other one is the somatostatin receptor, which picks up a neuroendocrine phenotype, which I think preceded the PMSA. But those are the only two, and the other antibodies don't seem to quite have made as much progress as those, but others may correct me. We're really interested in other examples. You need some yardsticks in the development of these probes, novel ones particularly, to measure yourself against, that have been tried in humans and work.
And my last question is, why use small molecules instead of antibodies to target tumors or bacteria?
Yeah, those are antibodies are large molecules. You can only put enough of those in to match your target. There's a stoichiometric binding restraint, means that the numbers of molecules you can add is lower. They won't enter cells very well. Small molecules, and I use the word small molecules a bit liberally. These are tethered molecules. The molecular weight's about 1,300 to 1,100 in our hands. Are smaller, they diffuse. You can use them at high concentrations, obviously. They act like drugs. So they have those advantages. And penetrating through a bacterial cell wall has to be small.
Thank you. Thank you so much for your time and for answering those questions, Dr. Haystead. We've reached the end of our session today, and to respect everyone's time, we'll wrap things up. As mentioned earlier, we will provide a written transcript of all the questions and answers in the coming days. Thank you, Dr. Haystead, for your presentation and for addressing our questions. We hope the information provided is relevant to your research. If you have further questions in the coming days, or if you're interested in any of our equipment, including the Super Argus, please reach out to us at Syntica. We would be delighted to discuss how we can support your research goals. Thank you all for attending today's session. We look forward to seeing you at future Syntica events. Have a great day, everyone.
Okay. Bye.